Today we're going to talk about membranes and lipid metabolism. So if you put a lipid into an aqueous environment, it tends to aggregate, and the type of structure formed depends upon the kind of lipid and the concentration of the lipid. There are three major types of structures that form micelles, liposomes, and bilayers. So let's look at each of these. So a micelle forms when the individual units are wedge-shaped. In other words, the cross-sectional area of the head is greater than that of the side chain. Here's a diagram of what a micelle looks like. It's something that forms with fatty acids, for example, or sodium duodecyl sulfate, SDS. In this case, each micelle would consist of a few dozen or up to a few thousand lipid molecules, and the aggregation occurs when the micelles achieve a concentration that's higher than a critical threshold. A vesicle or a liposome, on the other hand, is a case where small bilayers spontaneously seal into these kinds of spherical vesicles. In this case, there's a central aqueous cavity that can encapsulate some artificially inserted protein or drug molecule. So this can be used as a drug carrier, for example, and different vesicles can readily fuse with each other. Now, a membrane bilayer consists of two leaflets of lipid monolayers, shown here. In this case, the individual units are cylindrical, meaning the cross-sectional area of the head is about equal to that of the side chain. So the hydrophilic head group here can interact with water on the outside of the bilayer, and the hydrophobic tail is packed on the inside into this hydrophobic environment. One of these leaflets will face the cytoplasm, or the inside of the cell, and the other faces the extracellular environment, or the inside of an organelle within a cell. Now, there are a variety of functions of membranes. They define, in a fundamental sense, the boundary of the cell. This is kind of the primordial example of determining self versus non-self, what's inside the cell, what's outside the cell. When life emerged, the very first cells had to or existed to define that internal versus external aspect of self and non-self. Membranes also allow for import or export of things like nutrients and wastes and toxins. They allow cells to maintain metabolites and ions within the cell, or to import and export those kinds of species. And they also provide for compartmentalization, allowing you to separate different functions and activities within a cell. For example, to keep proteolytic enzymes away from key substrates that they could act on. And they can also, as you see in oxidative phosphorylation, store energy in the form of a proton gradient that exists across a membrane. Now, our understanding of membranes today is based on the fluid Mosaic model proposed in 1972 by Singer and Nicholson. And this idea is basically that lipids form a viscous two-dimensional solvent into which proteins can be inserted and integrated to varying depths. These integral membrane proteins are often associated with the membrane, typically spanning the entire bilayer, whereas peripheral proteins are only more weakly associated with the membrane, and these can be removed more easily. Some of these peripheral proteins are associated non-covalently through electrostatic interactions, and some can be linked to membrane lipids that allow for their tight association with the membrane. Here's a diagram of a fluid Mosaic model, and here you can see various phospholipid head groups, phospholipids, sterols inserted into the membrane. Here's an integral membrane, single-pass transmembrane protein. You can see some glycosylation and glycolipids in the extracellular environment. Here's a peripheral protein that's associated with the membrane, and here's a protein that's covalently linked to a lipid. Now, the composition of membranes critically can vary in different organisms, different tissues, and even different organelles within the same cell. A number of things can vary. The ratio of lipid to protein varies, the kind of phospholipids present in the membrane varies, the abundance and type of different sterols in the membrane varies. For example, cholesterol predominates in the plasma membrane of eukaryotes, but it's absent from mitochondria, whereas another example is galactolipids are present in plant chloroplasts, but are almost completely absent in animals. here's an example of how the membrane composition is highly variable within different organelles. So a good example of this is cardiolipin is a lipid that's only found in the mitochondrial membrane, not in other membranes like the plasma membrane, only a little bit in the outer mitochondrial membrane or lysosomes. Another key feature of membranes is that the membrane bilayers are asymmetric. So the inner leaflet and the outer leaflet have different lipid compositions. The outer leaflet is often more positively charged. Another important example here is phosphatidylserine. When it's normally present on the inner leaflet, but it can flip into the outer leaflet in platelets, which stimulates blood clotting. And in most other cells, when phosphatidylserine is found in the outer leaflet of the plasma membrane, that signals that the cell is undergoing apoptosis and marks the cell for destruction. That phosphatidylserine exposure is the so-called eat me signal that triggers the engulfment of that cell by other phagocytic cells. So here are some examples of lipids that are found asymmetrically in the inner leaflet versus the outer leaflet of membranes. Phosphatidylethynolamine, for example, found on the inner leaflet, and phosphatidylserine, which we mentioned. There's a number of other examples here. Now, the proteins that are embedded within membranes have particular functions. So receptors, for example, detect signals from outside the cell. These can be hormones or neurotransmitters or pheromones. These proteins in membranes can act as channels, gates, and pumps, and as enzymes involved in lipid biosynthesis or even ATP synthesis in mitochondria. One way that proteins can be associated with membranes in a reversible fashion is through so-called farnesylation or lipidation. Prenylation is another term for that, attachment of prenyl groups. So in this example, proteins can be attached to the inner leaflet through attachment of a farnesyl group. The signal that determines whether a protein is farnesylated is a C-terminal 4-amino acid sequence known as a CAX box. C stands for cysteine, A is a small aliphatic amino acid, and X is methionine, serine, glutamate, or alanine. So if a protein has this CAX sequence, then it's very likely to be a subject of the enzyme farnesyltransferase, which transfers the C15 farnesyl lipid onto the protein, which causes it to be associated with the membrane. Okay, let us turn now to how lipids are actually degraded. So how do you take the membrane lipids and use them for harvesting energy? How do we catabolize lipids, in other words? Now at this point I'm going to take a pause and I'm going to show you this video.
Those fats, 'cause I need some energy that lasts. With 3 fatty acids and glycerol, give me just what it takes for the long haul. These are the macromolecules,
Okay, so now we'll go back to our PowerPoint and look at how our membrane lipids degraded now. So lipids can be used as a tremendous store of energy. A good example of that is hibernating bears get the majority of their energy during their long winter months of hibernation from stored fatty acids. So fats are so useful because compared to polysaccharides, there's a lot more energy per carbon because fats start out being completely reduced to a large degree, whereas carbohydrates are already partially oxidized. And in addition, another key factor is that fats carry less water. They're nonpolar, so when they're stored in the absence of water. So glucose and glycogen, for example, can be used for short-term energy needs, but fats are very useful for long-term energy needs and are a good place to store energy for this long-term usage. Now when you break down a fat starts in the form of a triacylglycerol. That's a triester of 3 fatty acids and the trihydroxyl molecule glycerol. So when you break down this triacylglycerol into the free fatty acid and the glycerol molecule, that's a reaction that's catalyzed by various lipases or esterases. Now these lipases are often regulated, for example, by the hormones glucagon and epinephrine. Epinephrine acting as a signal saying we need energy now, rapid mobilization of energy, and glucagon is a signal saying we're out of glucose, time to mobilize more energy. So here's a depiction of a long-chain saturated fatty acid and a fatty acid with a single double bond in the cis orientation. Now the glycerol that's released from a fat upon hydrolysis of the ester of the triacylglycerol can then enter glycolysis. So glycerol gets phosphorylated to make glycerol 3-phosphate. That can be oxidized to make dihydroxyacetonephosphate. That should be familiar as an intermediate in glycolysis. The enzyme TIM, remember, can act on dihydroxyacetonephosphate and convert that to glyceraldehyde 3-phosphate, and that goes right into glycolysis in the normal way that we saw when we looked at glycolysis. So because of this, glycerol allows for a limited amount of anaerobic catabolism of fats, in other words, extraction of energy from fats without the use of oxygen. The activation, the degradation of the fatty acid component, as we'll see, requires oxidation, but for the glycerol catabolism, that can occur through this glycolytic pathway, which doesn't require oxygen. So here are those reactions. Glycerol is phosphorylated first into glycerol 3-phosphate, shown here, uses 1 ATP, and then there's an oxidation reaction to get to dihydroxyacetone phosphate. So that's an NAD plus dependent reaction, oxidizes this alcohol to a ketone by elimination of this hydride in the usual way, which is placed on NAD plus, generating NADH. That dihydroxyacetone phosphate now, just like in glycolysis, can be isomerized to glyceraldehyde 3-phosphate through the enzyme TIM, triose phosphate isomerase, and then the rest of the reactions of glycolysis proceed. Now for the rest of the fatty acids that are generating in the mobilization of fats, these have to go through a different mechanism that we're going to focus on here. The fatty acid has to be broken down into a sequence of acetyl-CoA units that feed into the TCA cycle, and then the energy is extracted just in the same way that we saw when we looked at the TCA cycle in the context of carbohydrate metabolism. So in other words, the acetyl-CoA units go into the TCA cycle, we extract electrons in the form of NADH and FADH2, and those go into the respiratory chain for the synthesis of ATP. So the key question is, how do we oxidize the fatty acid and generate these acetyl-CoA units? So this is a pathway known as beta oxidation. Each time we run through this cycle, we generate one acetyl-CoA unit. And it's really a set of reactions we've seen before. Exactly the same set of reactions are seen in the TCA cycle. So we start with a fatty acyl-CoA, and we do a dehydrogenation of the alpha and beta positions relative to the thioester. That introduces a double bond. That's an FAD-dependent reaction, so we're getting FADH2 as a product. Now we have an alpha-beta unsaturated thioester. That's a good Michael acceptor, so we can do an addition of water to the 4 position here, generating a beta-hydroxy fatty acyl-CoA, and now we oxidize that in the usual way with NAD plus to generate a beta-keto thioester, and then we can do a carbon-carbon bond cleavage reaction catalyzed by thiolase, where we eliminate one acetyl-CoA unit and shorten this fatty acyl-CoA by 2 carbons. So every time we run the cycle, again, we release one more acetyl-CoA and shorten the chain by 2 carbons until we get down to a single acetyl-CoA unit. So let's look at those reactions again. First step, dehydrogenation of an alkane to make an alkene. This occurs on the inner mitochondrial membrane, and this is very analogous to succinate dehydrogenase in the TCA cycle. The second step is hydration, so addition of a water molecule to the double bond that we just introduced. And again, this is analogous to the fumarase reaction in the TCA cycle. So let's look at those 2 reactions here. First is the dehydrogenation reaction, where we eliminate, we introduce a double bond here in the alpha-beta position, FAD dependent, and then the next step is addition of a water molecule here to the 4 position, creating a beta-hydroxyacyl-CoA. Now steps 3 and 4 is oxidation of that alcohol to make the ketone, and then the cleavage, the carbon-carbon bond cleaving reaction. So let's look at those 2 steps shown here. First, the oxidation of the hydroxy group to a ketone in an NAD-dependent manner. So now we have the beta-keto-acyl-CoA. And then here's the key carbon-carbon bond cleavage reaction. So the way this works is that the thiol of coenzyme A adds into the ketone, makes a tetrahedral intermediate, and when that collapses, we kick out this acetyl-CoA unit as the leaving group. So if we look at a fatty acid like palmitic acid, C16, we do this process 7 times. We cleave off 7 acetyl-CoA units. We're left with the 8th one, the last one. And so we end up with 8 molecules of acetyl-CoA, 7 molecules of FADH2, 7 molecules of NADH, and then the acetyl-CoA that's formed can enter the TCA cycle, be further oxidized into carbon dioxide to make more GTP, NADH, and FADH2. And so again, we break the chain down by 2 carbons in each cycle, as shown here. And a similar set of mechanisms are used in a variety of other metabolic pathways where we have this sort of prototypical sequence of dehydrogenation to introduce a double bond. We see that in the TCA cycle, for example. Also the oxidation of some amino acids. That gives us a double bond. Then we add water to add this hydroxyl group in. And then finally the NAD-dependent oxidation of that alcohol into a ketone. So these 3 sort of prototypical reactions are seen again and again. Now another key aspect of fatty acid degradation is the possibility of forming so-called ketone bodies. So if acetyl-CoA is to enter the TCA cycle, we need oxaloacetate as the acceptor for acetyl-CoA. When oxaloacetate has been depleted, the acetyl-CoA is instead converted into ketone bodies that frees up coenzyme A so we can do continued beta oxidation of fatty acids. So the first step in the formation of ketone bodies is just the reverse of this last step in the beta oxidation pathway, which is to regenerate acetoacetyl-CoA. So let's look at that reaction here. We take 2 acetyl-CoA molecules, put them together in this reverse reaction, and now we get acetoacetyl-CoA. Now, we could imagine just hydrolyzing this thioester to make acetoacetate, which is the final product here, or the penultimate product. But in fact, nature does this in a slightly more complicated way, brings in 1 more acetyl-CoA unit to make this intermediate, the HMG-CoA, which is a key intermediate in lipid biosynthesis, as we'll see in another video. But the key point here is then we can go from there to acetoacetate by elimination of one of these acetyl-CoA units. And now acetoacetate is a subject for 2 different possibilities. It can either be decarboxylated to make acetone, a so-called ketone body, or it can be reduced to make beta-hydroxy butyrate, shown here. So we just reduce this ketone to make the alcohol. Okay, so to summarize what we looked at in this video then, lipids are critical for forming membranes. The lipid content of membranes is highly variable and functional in different organelles and different cell types, and lipid catabolism involves a sequential breakdown of acetyl-CoA units. Great, I look forward to seeing you in class.